Okay, good afternoon. My name is Sasha Ames. I'm a postdoctoral researcher at Lawrence Livermore National Lab. There's the mic. Okay, so this is some work I'm doing on this metagenomics search application research team. Some of the colleagues are listed up there, looking at efficient k-mer indexing. So to introduce this particular problem space, yesterday's talk discussed a little about metagenomes and microbial genome samples. So it's the study of microbial communities found in environmental samples. So this is, as opposed to sequencing genomes from a single organism, it's a heterogeneous sequencing from potentially many organisms. So the goal in analytics is to one particular goal is to figure out what organisms are in a sample. So there are many crucial applications of this, like in clinical diagnostics. Imagine you can swab a patient's cheek, sequence it, and figure out what infects the patient as opposed to running a battery of tests. And there's some other interesting problems recently that have come up where this could be applicable. So sequencers are advancing in performance. They're getting faster all the time, while the computation, that's specifically the analysis necessary to process the sequencing, is lagging in terms of capability. And with the most standard tools for this are sequence alignment tools, and there's a trade-off in speed versus accuracy. The faster tools tend to be less accurate, and highly accurate tools tend to go very slow. Also scalability limitations. Also, it's very beneficial to put analysis close to the sequencer. So imagine you have a node attached right to a sequencer as opposed to having to ship the sequence data out to a supercomputer center for analysis and then ship back the results. So I'll talk about what we're doing about this problem. That's the Livermore Metagenomics Analysis Toolkit. So this is a more database-oriented approach. We're not using any particular platform. We're doing our own indexing. So we base an index of subsequences of length k called k-mers. And each k-mer refers to a constituent organism. And using this database, we can do classification. And given the time constraints, I'm not going to talk about the classification algorithm. So the index size seems pretty paltry at 300 gigabytes, but what it is about that size actually has 19 billion keys, and given our requirements for indexes, very low latency, we want to get tens of millions of lookups per second in order to do efficient classification. So of course, ideally you'd want to fit this database in memory, but memory is expensive, so maybe we can use flash as a lower cost alternative. So as far as indexing goes, if you want to use flash, you want to reduce the number of page fetches. But the advantage we have for this application is, well, we can make a read-only index where we don't worry about updates, and we don't need range queries. So we have an in-house solution for indexing. We call it the two-level approach, for lack of a better term. So a lot like a hash table, but the idea is to deal with the chaining by using a second level, and you put all the items in a single list. So every k-mer you can encode as an integer, and that makes our life easier rather than dealing with variable length strings, and you can split the integer. So the first level, which is basically the table, takes the first part of the integer key and maps it to the second level, which these lists fit on a single page of memory, so you can fetch it and then do a quick binary search. Now there's a trade-off in how this is organized because it's not clear how to organize these lists. So we've sort of made that determination by level experimentation, but ideally you fit the first level, make that small enough that that will fit in memory, so you're just fetching pages for the second level lists. So not much time left, but I'll just look a little at performance, comparing our index with that of off-the-shelf hash table as GNU hash. And one thing we looked at is, well, when you give it more memory for cache, does the performance improve? And we find the blue curve, or the optimal configured two-level hash does improve while regular hashing doesn't. And I'm out of time. Thank you.